Okay, in this video, I just want to talk about geometrical isomerism, EZ isomerism, and how it can be fairly important in biology sometimes. Okay, now this molecule here is actually it's called retinol. Okay, and that is all. It's got an alcohol group. It's vitamin A. Okay, now vitamin A, you probably have heard the old wives' tale. You've got loads of vitamin A in carrots, and say, oh, if you eat carrots, you'll be able to see in the dark, which isn't true. But there's an element of truth in it, in that you do need vitamin A in the rod cells in your retina. Now, you can see here there's quite a lot of double bonds in that molecule. But we need to look at this one here, right? And I'm going to put the invisible hydrogens that you can't see. So there'd be a hydrogen there, wouldn't there? And there would be a hydrogen there. And you can probably appreciate that this double bond, the arrangement around here, right, is the Z form. It's the Z isomer. In old-fashioned terminology, that would be called the cis form. Okay, now in your retina, this is attached to a protein here. So the alcohol group is attached to a protein. So the important thing is that having the molecule in the Z form, you've got a kink in the molecule. So the molecule isn't straight; it's got this kink in it. What happens is if light falls on your retina, what happens is it will break the pi bond, and that allows, and then so the light, you need energy to break a pi bond. It's coming from the light. You break the pi bond, and the pi bond can then reform, and it won't be in the Z form. It will form here in the E form. And you'd probably be able to appreciate there, if that happens, the molecule, the kink that's in the molecule is going to disappear. So I'll just rub that out and try and draw what happens then. Okay, so you're getting light. What's going to happen is it's going to become this shape instead. Okay, I won't put all the other bits on, but you can see it goes from there to there. Now this end is attached to the protein, so the protein is going to move, and that then sends a nerve impulse. Okay, so what happens is you have cis, let's call it the Z retinol, which is sometimes called cis. You get light, and it converts it to E retinol. That's sometimes called the trans form. And when that happens, you get movement, and you get a nerve impulse is triggered. Okay, so it sends a message down the optic nerve, and that is perceived by your brain as being light, and that's how you see. Now you may have experienced that when you go into a dark room, at first you can't see anything, and after being in there for about 10 minutes, you can start to see more and more detail, and we can explain that. And it's not that phenomena of your light becoming dark adapted is not due to the pupil dilating, because that occurs almost instantaneously. It takes like 10 minutes. And what's happening here is like, right, and you've got bright light. In bright light, you're going to have so when it's bright, you'll have low amounts of the Z form, because it's all being converted to the E form. Okay, now obviously to enable you to see, that has got to be converted back. The E retinol has to be converted to the Z retinol. That requires ATP. It takes a bit of time, And so when you go into a dark room, the E-retinol is gradually converted back to the Z-retinol. You've got much higher concentrations of Z-retinol, so your eyes become more sensitive to low levels of light. Bright light, you haven't got so much of the Z form, so it's not so sensitive. You probably, you burn a piece of magnesium ribbon, you look at it, and you have a big white blob in the middle of your eyes for a minute or so, and that's because all of the Z-retinol there has all been used up and turned into E-retinol. You can't see anything for a bit there. Okay, so transformation from the Z form to the E form is really important in being able to enable you to see. Right, let's look at another important thing. So what I've got here is, you can probably see that they are, what functional group we've got here. We've got a carboxylic acid. Okay, this is actually called oleic acid. It's 18 carbons long, okay, and it's a very common fatty acid that you'll find. Okay, now all of the this is the Z form, this one here. So you see these invisible hydrogens are on the same side, okay, whereas this here is the E form. The invisible hydrogens here are on opposite sides. That's the E form. Now, naturally occurring fatty acids with a double bond, they are always in the Z form. So it's 100% in the Z form. You don't get the other isomer naturally occurring at all. And that's important because it means this fatty acid has got a kink in it. Now, if you do biology, you probably know that fatty acids are involved in phospholipid molecules. Okay, so I will draw a phospholipid molecule. Basically, it's got this green bit, which is it's got a phosphate group and a nitrogen containing thing in it. And then you have two fatty acids. Okay, so I'll draw one. It should be 18 long or 20 long. That one is a saturated one. Okay, and then this one is going to be unsaturated. And that's important because what happens is, where the double bond is, we have a kink in it. Okay, so this is the this in biology we would call this the this is the hydrophilic head, isn't it? And that sits outside. And this is the hydrophobic tails. And here we have got a double bond. That's an unsaturated fatty acid, and it produces a kink in the molecule because it's always in the Z form. Now, how does that affect the phospholipid bilayer? So if I draw the phospholipid bilayer, you're going to have, you've got your phospholipids here, yeah. I'll just do one more, okay. And so this is outside the cell. This is the cytoplasm here. And this is the cell membrane, yeah. Right, let's imagine we have one of these now which contains an unsaturated fatty acid. So instead of being straight lines like that, this one is going to be like that. Okay, so that means the one next to it can't pack quite as close, can it? Okay, so that means, because there's not so many points of contact, so you get reduced van der Waals forces. Yeah, the van der Waals forces between the one with the double bond in it are slightly weaker. Okay, and that means the membrane is more fluid. Because the intermolecular forces between the phospholipid molecules are weaker, and it's important that the membrane needs some fluidity, so it needs to have some of the phospholipid molecules need to have double bonds in them. Also, in cell membranes, you tend to have cholesterol molecules which insert somewhere between the phospholipids like this from time to time, and cholesterol reduces, that also reduces the van der Waals forces, and it makes the membrane more fluid. So, as you know, cholesterol, blood cholesterol levels, how much unsaturated fat you have in your diet, those two things are interrelated. Yeah. You probably also know that saturated fats tend to raise your blood cholesterol, unsaturated fats tend to lower the blood cholesterol. And you may have heard of this. Now, this is the E form. The old-fashioned form of this, you'd call this the trans form. And you may have heard that these so-called trans fats don't occur naturally, but you get them when you when unsaturated vegetable oils are hydrogenated to make them saturated, and you get this contaminant essentially produced with unsaturated fats in the trans form. Now these are straight and they are rigid, and they're extremely bad for you. They're very much implicated in raising blood cholesterol levels. Okay, and it's because their shape they're not the correct shape. They don't have the kink in them. They're straight, and that has an effect on membrane fluidity. Okay, so just a little bit of biology there. We'll leave that.